So I'm going to be talking about nerves and specifically objectives 6.5.2 through 6.5.6. Okay, so the first objective is 6.5.2, and the objective is to draw and label a diagram of the structure of a motor neuron. So I got this image off of the internet. However, you don't need to know all of this for the test, even though you can learn it and it's not going to hurt you. But the things you do need to know is where the nucleus is, which is right here. And then you have your dendrite, your soma, which is the body, and then your axon, which is this, and then you have your motor end plate, and you just need to know where the signal is going to end here. And then you can also know what a Schwann cell is. And then you need to know the direction of the nerve impulse. However, if you know that this is the motor end plate, then that won't be an issue. And that's all for that objective, so pretty much you just have to memorize this. Okay, so the next objective is state the nerve impulses are conducted from receptors to the central nervous system by sensory neurons, within the central nervous system by relay neurons, and from the central nervous system by motor neurons. So first you need to know what the three things are. So sensory neurons conduct nerve impulses from receptors to the central nervous system through the afferent pathway. Relay neurons conduct nerve impulses within the central nervous system, and these are also called interneurons or connector neurons, as you can probably see from the diagram as to why. And then you have motor neurons that conduct nerve impulses from the central nervous system to the effectors. So in order to see how this works, you first need to know what a stimulus is. All scenarios that require a response go through a stimulus response pathway, and the stimulus is the first step of this. A stimulus is created by a change in your external and or internal environment, and this triggers the stimulus response pathway. So for example, if you touch something very hot, it's going to stimulate this response pathway. Receptors are the next part of this pathway, and they are specialized structures that respond to a specific type of stimulus. They initiate signals by triggering either the nervous system or the release of hormones, and the receptor sends the signal down the sensory neuron to the central nervous system. And that's next. And then you have your effector and your response. So the central nervous system then sends a signal through the relay neuron and then through the motor neuron. So this relay neuron only acts within the central nervous system, and then a signal goes through the motor neuron, which goes to the effector. And the effector is the organ that receives this signal. And the response and the action that occurs when the effector organ reacts to the signal from the control center. And the response changes the external and or the internal environment to stabilize the initial change. Okay, so that's all for that objective. The next one is to define resting potential and action potential, which are depolarization and repolarization. These are the 4 definitions that you need to memorize. So resting potential is the charge difference across the membrane when the neuron is not firing, and this is maintained through the sodium-potassium pump. The action potential is the charge difference across the membrane when the neuron is firing. Depolarization is the change from a negative resting potential to a positive action potential, and this is caused by the opening of the sodium channels to pump inwards. And then the repolarization is the change from a positive action potential back to a negative resting potential, causing the opening of the potassium channels to pump outwards. The next objective is to explain how nerve impulse passes along a non-myelinated neuron. So first there's a resting potential, and this is where the sodium-potassium pump maintains an electrochemical gradient of the resting potential. And this pump is a transmembrane protein that uses active transport to exchange sodium and potassium ions across the membrane. And it expels 3 sodium ions for every 2 potassium ions emitted, so this makes the inside of the cell membrane relatively negative in comparison to the outside of the cell membrane. So if you can imagine this, it's just potassium being pumped in and sodium being pumped out at a constant rate. Then there's the transmission of an action potential. So sodium and potassium channels in a nerve cell are voltage gated. So this means that they can open and close depending on the voltage across the membrane. And in response to a signal at a sensory receptor, dendrite sodium enters the neuron passively. So this is where the pump almost switches and sodium enters the cell instead of exits it. And this influx of sodium causes the membrane potential to become positive, and this is what depolarization is, as was defined before. The threshold potential is achieved when the threshold potential is achieved, adjacent voltage gated sodium channels open, generating a new wave of depolarization, which is the action potential, and this is going to spread down the axon. This change in membrane potential also activates the voltage-gated potassium channels, which causes potassium to exit the neuron, which is backwards of the sodium-potassium pump once again. And this efflux of potassium out of the neuron causes the membrane potential to become negative again. And that is your signal, I guess, and in order for the neuron to fire again, the resting potential has to be reached, and thus the pump has to be reestablished. So here's an animation of it, and the threshold is that dotted red line you see in the diagram on your left, and in order for the action potential to be reached, the feeling has to be big enough to pass this threshold. So once it gets past that, then the action potential is released, but this is a really good animation of what exactly happens. Okay, so next you have the principles of synaptic transfer. This is our last objective. So to define two terms first, a synapse is a junction between two neurons. It's like the little space in between neurons, actually, and then action potential, it can't cross the synaptic gap, so it triggers the release of chemicals, which are neurotransmitters, to continue this signal to the next neuron. So the chemical transfer across synapses, this happens when an action potential reaches the axon terminal, which is the end of the neuron, and it triggers the opening of the voltage-gated calcium channels. So this calcium diffuses into the cell and causes the vesicles to fuse with the plasma membrane, which then in turn allows neurotransmitters to be released from the axon by exocytosis and to cross the synaptic cleft. Then the neurotransmitter binds to an appropriate neuroreceptor and on the postsynaptic cleft, and if it's an excitatory neurotransmitter, the reaction is going to continue. However, if it is inhibitory, the reaction is going to stop. And this is a good animation of it once again. It just shows like what exactly is happening at every single point. And that is it.